Copyright Stanford University, all rights reserved.
I will allow the introductions for our guest speaker, Dr. Weissman, very well known to all of you, I hope, by Ingrid Park.
So it's a pleasure to introduce today's speaker. He is a prolific writer. As of this morning, PubMed shows he has over 700 research articles dating back to 1961. He has shaped the entire stem cell field, and his mentoring style reminds me of a Chinese proverb, which I will read. Give a man a fish, and you feed him for a day. Teach a man to fish, and you feed him for a lifetime. So, or teach us how to fish.
And good. So, to remind you, the difference between stem cells and other cells in any tissue is incredibly simple. When stem cells divide, they can give rise to more stem cells, as well as differentiate to give rise to cells which, by quantum steps of differentiation, will give rise to the tissue. So blood-forming stem cells self-renew to make stem cells, but also give rise to blood. All of the steps between stem cell and blood are non-self-renewing cell divisions. So the entire activity of the blood, its development, its maintenance, its transfer in a bone marrow transplant, and when things go wrong, are really dependent on that one cell type. Now, we study blood-forming and other tissue stem cells in vivo because we found, as many people should have found, that trying to study it in vitro leads to artifacts where you think you're going down a stem cell path, blood to make heart cells, which they never do, mesenchyme to make heart cells, which they never do. So in order to avoid those mistakes and wasted money and dangerous clinical trials, it's important to purify the cells and test them in vivo. This should go. There we go. So we isolate stem cells with many antibodies, and at the end of the day, we can get from mice and from humans purified stem cells in a tube. That means we can get rid of cancer, the blood-forming tissue from a patient who has metastatic cancer. And that means we can get rid of T cells if we need to transfer stem cells from a donor to a host without causing an immune reaction of the T cells against the host called graft versus host disease. Many years ago, at a company I co-founded, had stock in, but it's gone now, Systemics, we did a clinical trial trying to metastatic breast cancer, their stem cells free of cancer cells. And we could do that. We got a 250,000-fold deletion of cancer cells, breast cancer cells, from mobilized blood on its way to be giving you pure blood-forming stem cells. So we did a clinical trial at Stanford led by Rob Negrin and Judy Shizuru, and we found in retrospect that our therapy of cancer-free stem cells led to 33% overall survival. Now it's over 15 years, over 16 years, whereas all of the women who got mobilized peripheral blood back, but they were told they were getting stem cell transplants, but they had contaminating cancer cells, at least 50% of them, in the transplant that went back into their body after they received fully myeloablative therapy in an attempt to kill the last cancer cell in the body, only had 7%, and even those had cancer, those few in the 7%. Now that's statistically significant at all stages for both overall survival here and progression-free survival. So when people tell you you need thousands of patients to power a trial, that's if you're looking for a tiny difference. If you're looking for a big difference, if you're lucky enough to see it, there were 15 patients in that group, 74 patients in that group. So, of course, if you know somebody who has metastatic breast cancer, you would say, where's the therapy? And the therapy is gone because the company that bought Systemics and held all the intellectual property made a business decision in the year 2000 to close down all of the stem cell and stem cell related programs without giving back the stem cells to the founders or the university. That's not a rare occurrence. So one of the most important things for us to think about here at Stanford is how do you extend the time when you go from academics to commercialization? Because obviously, if you had seen these results, even at this point, you would say this is something that's worth expanding and understanding and delivering to the vast amount of people and trying to find cheaper and cheaper ways to do it. But none of that is happening because they made a business decision. They got a pill that they could hand to people that would treat them for another disease for life rather than this curative intent therapy. Now, the other use of blood-forming stem cells, perhaps what will be the most important use, is to regenerate damaged or genetically defective blood-forming systems. So in that case, having T cells as well as stem cells in the graft leads to graft versus host disease. And if you go to the transplant ward, you will see that those patients who have allotransplants are in serious condition. And it is the art of medical practice of bone marrow transplant, of hematopoietic cell therapy, that lets people survive, and the graft versus host T cells help treat the disease they have, usually a leukemia, but sometimes a lymphoma. But if you have sickle cell anemia or severe combined immunodeficiency or beta thal, the last thing you worry about is getting rid of all of the cells with the T cells. What you need is simply to replace a genetically defective system. We have shown mouse to mouse, human to human, mainly with Judy Shizuru, human to the skidoo mouse, that pure stem cells, no matter what the dose, cannot cause graft versus host disease. And when they engraft, we've shown also and published many times in the 90s, they induce permanent transplant tolerance of heart grafts from the donor, pancreatic islet grafts from the donor, skin grafts, with no other immunosuppression. So this would be the platform for regenerative medicine if we could make it safe and easy. But of course, the company owned all this. We now have the rights back to separate stem cells at Stanford. Judy Shizuru and Maria Grazia Roncarollo will be leading the charge to move it step after step in an academic rather than a commercial setting, although some people think the hospital might be interested in how much money they can make. Judy and I also showed that you could reverse mouse type 1 diabetes, the autoimmune attack, with stem cells, blood-forming stem cells from donors that are genetically resistant to getting type 1 diabetes. And then we went on to show it works in lupus. And Judy has data, I haven't seen the published data yet, in a mouse model of multiple sclerosis. So now you could imagine this is no longer a minor field. Organ transplants, immunological tolerance, repair of genetic diseases, all with one cell type, held back by a company. So where else might we get stem cells? And I'll come back to this later, but we know, and this is why Prop 71 was passed, that embryonic stem cells, which are derived from the inner cell mass of the preimplantation blastocyst and cultured in vitro so they retain embryonic fates, pluripotent fates, can give rise to all kinds of tissue cells. And we believe, and we hope we'll find it in enough cases, that they must pass through blood-forming stem cells to make blood cells, brain-forming stem cells to make brain cells, and so on. So these might provide a source of cells for regenerative medicine eventually. That is, that if you could find the fates of the mesoderm germ layer, you ought to be able to make bone or cartilage or heart cells or skeletal muscle or blood. And in a much less crowded room, the last hour, Roger Peterson was talking about exactly that case. So we need to know what the fates are in vivo of the various parts of the mesoderm that give rise to bone, cartilage from this fate, skeletal muscle, and if you need a little brown fat, this, cardiac mesoderm, and off another branch, blood-forming cells. So, for us to get there, we have to have tissue stem cells. But what I didn't tell you very much is that all of the transplants that are done today of bone marrow or mobilized blood start with near lethal or lethal irradiation or chemotherapy of the host. So we need to get rid of that. We propose to do that by removing blood-forming stem cells and established T cells and established natural killer cells with antibodies rather than drugs. So that in the far future, we might be able to use embryonic or induced pluripotent stem cells as sources of both the blood-forming stem cells that will induce tolerance to any other tissue or cell from that same ES donor, which could give rise to neural stem cells or cardiac precursors and so on. To do so, we began, Anishka Chekovich, Tita Bhattacharya, and I, to look for antibodies that in the mouse would remove blood-forming stem cells selectively so that the animal wouldn't die of other problems that irradiation, for example, could cause death. And so we looked at one antibody against blood-forming stem cells that was a function-blocking antibody for a tyrosine kinase receptor on blood-forming stem cells called c-KIT. Shinichi Nishikawa had made that. And so when we transferred 5,000 highly purified mouse blood-forming stem cells without radiation or chemotherapy into severe combined immunodeficiency mice, that is, that lack T cells, B cells, and NK cells, even if you gave 5,000 cells, which is 50 times the dose needed to save them from irradiation, you only got 1% donor-derived cells. But a single treatment with that antibody, and it was 15%. And if you did that 3 times, antibody, then stem cells from this donor, then antibody, and then stem cells a week later from this donor, then antibody, then stem cells, again, from this donor, by the time we look 6 months out, it is a tetrachimera, you could say, with 3 different kinds of blood plus a little bit of host blood. But you can see that now we're at about 80% chimerism. That cures sickle cell, severe kind immunodeficiency, beta thal, alpha thal, all of the diseases we're talking about, sufficient to induce tolerance. So this is where we want to go. And I'll just say that we are now looking at antibodies that might synergize with the function-blocking ACK antibodies, we being Ankasha Chabra, Aaron Ring, Jens Volkmer, Kip Weisskopf, me in a minor role, Judy Shizuru. And we have now got antibodies which synergize with that to lead to donor-derived chimera. And we'll be looking not only at this function in mice, but Judy Shizuru is already lined up to do a clinical trial here at Stanford with children with severe combined immunodeficiency who will receive a function-blocking human anti-human c-KIT followed by highly purified blood-forming stem cells. So they have a little bit of a time to convince the FDA that they can do 2 variables at once, but we'll see. At least it's back and starting. Now, I'm going to get back to this issue in a minute. I wanted to move this slide, but I wanted to say that we found when we were looking at blood-forming stem cells that, in fact, as we age, we change the kind of stem cells we have. They're mainly myeloid biased when you're old, that is my age, and mainly balanced to make lymphoid and myeloid cells when you're young. And we came to the conclusion, before we ever looked at this, that this could be a diversity of stem cells that are present at birth, the myeloid bias being present rare at birth, and the balanced one becoming rarer in old age, but that no single stem cell would change its function, but we would be looking at stem cell competitions, okay? And that over time, these are better fit in old bodies, and these are better fit in young bodies. But how did we even start thinking about that? Oh, please. Oh my God. It was really out of place. Keep the thought for a second. When we were looking for other tissue stem cells, we looked at brain first. Rusty Gage, David Anderson, my wife, and Tsukamoto Weissman, and I founded a company, 1997, so that's before the disaster happened with the other company. It wasn't a financial disaster. And so we found that we could isolate from human fetal brains cells with a bunch of antibodies, which were highly purified and at the single cell level could make a sphere of neural cells which contained the capacity, when transplanted into the brain of immune deficient mice, of giving rise to neurons, astrocytes, oligodendrocytes, and if you do it early enough, even the kinds of neurons that are present in the cerebellum. So all of these brown spots came from one injection of human neural stem cell-derived neurospheres into the lateral ventricle. And you can see the concentration. This is 47 weeks later. So one of the characteristics of mouse neural stem cells and now human brain stem cells in the mouse is that they can migrate. And they migrate after division in the neurogenic zone. There are two neurogenic zones in the adult brain, the subventricular zone, the dentate gyrus of the hippocampus. Most of us, as we get old, have fewer and fewer cells dividing in the dentate gyrus, and that's why we can't remember why we went from one room to the other. Or we can't remember the name of somebody we thought we knew pretty well. We remember everything from the deep past, but we can't make fast associations. These human cells seed both the subventricular zone and the dentate gyrus, and a year later, in the niche for stem cell division, the human cells are dividing along with the mouse neural stem cells. In the migratory path to go to the olfactory bulb, they are packing that pathway. They're not as efficient as mouse, so they're probably going slower and make up a higher percentage of the migrating cells. When they hit the olfactory bulb, they go just like the mouse neural stem cell derivatives. They gave rise to mainly GABAergic, but some dopaminergic neurons that are in the periglomerular zones. This is the DAPI stain, so you can see where the cells are. And they send their processes toward the olfactory glomeruli that are receiving input from the nasopharynx odorant and pheromone receptor cells. Okay? That continuous process is going on. So now that we knew we could give rise to neurons, astrocytes, and oligodendrocytes, we wanted to test the cells in a clinical trial. So the clinical trial was done with Eileen Anderson, Brian Cummings, mainly carried out by Nobuko Uchida, who is at the company we co-founded, Stem Cells Inc. I still have stock in it, so you might think I'm biased. But if you look at the value of the company, you won't think I'm biased. They did compression of the 9th vertebrae, thoracic vertebrae. this leads to a compression that leads to ischemia in the spinal cord. The cells that have their cell bodies in that region die of ischemia, but the motor neurons above and the sensory neurons below don't. But they're sending long tracks through the region where the oligodendrocytes died, and so they're demyelinated. Most of the consequences of spinal cord injury that are not slices of the spinal cord are due to demyelination, most. So these human cells were put above and below this lesion in an immune deficient mouse, and you're seeing the spread of the daughter cells. Very intense around the long tracts that are still barely intact at the edge of the spinal cord. But the consequences of this are that those that got neural stem cells were recovered in their hind limb paralysis and sensitivity. So they were no longer paraplegic, walking perfectly normally. Highly significant at all those time points. But those that got mesenchymal stromal, some people call them stem cells, the cells just sat in a bubble and did not regenerate activity sufficient to walk. Yet the vast majority of people in the world who are getting cellular therapy for spinal cord injury are getting which one? Are you listening? Say it. Mesenchymal. And they're spending $85,000 to $150,000 to get it. And it's not fixing the paralysis. So there's something wrong here between the academic and the medical culture and the commercial side. You shut off that which works, and you expand and sell at a high price that which doesn't. Aren't you a little worried about this? When we give diphtheria toxin at a dose that kills the human cells, they're immediately demyelinated and paralyzed. So the cells we put in were not only necessary for repair, but they were necessary for the maintenance of the function. Look carefully to see if anybody else who claims they have cells that repair spinal cord injury have the same sort of thing. The trial has gone on. Raphael Guzman, formerly a neurosurgeon here, went to Zurich Baldrist Hospital, and they carried out a trial of, I think, 15 people with Asia A. Half of the people with Asia A paralysis who had absent stimulation response feeling to impaired but present stimulation response. They didn't get up and run like in the movies, but they improved, including one patient who reported that now he could tell when he was going to have a bowel or bladder movement. That changed his life considerably. So that is moving to clinical trials in the US. I believe one part of it will be done here at Stanford, but since you have to separate yourself from the business and the academic side appropriately, I don't know for sure. Now, competing stem cells. Where could we have thought there were competing stem cells? Many years ago, I began studying organisms which separated from the rest of us at about 550 million years ago, if you believe somebody was there to record it. But, you know, it's reasonable to speculate that the dating is probably right. These are protochordates. I studied them because they have a natural transplantation reaction in vivo. Pardon me, in nature. So that you can see that they, as part of their lifestyle, will have immune rejection. This is the organism. In the state you mainly see it. It's a rosette of individuals under a gelatinous tunic. They have blood vessels. They have a heart, two-chambered heart. They have gills. They have a gastrointestinal tract with an oral and anal siphon. They have a whole bunch of things, including 11 different blood cell types. They send these vessels out so they have both extracorporeal and intracorporeal blood vessels. And at the end, they form little bulbs that are important. This colony of genetically identical clonal progeny from a single organism actually came after sexual reproduction through the steps that we went through, blastula, gastrula, and urula. Formed a tadpole which had a notochord, neural tube, segmented musculature, head, tail, eye, brain. Swam from the mother colony, it's a marine species, just subtitle, to another subtitle surface, lost all the chordate characteristics, formed this individual, but now, in order for this individual to make that colony, they begin to bud cells out of the body wall, which, as I will show you, or you'll have to believe me, are stem cells for the tissues and organs. So they have both the reproductive pathway to make an organism and an asexual reproduction, which is akin to repair of tissues by tissue-specific stem cells. When we looked at the gene expression of their blood cells and other cells, we found this is just us, them, lower to higher, that this organism is the closest to the vertebrates around for the gene sequences of related genes. So it's reasonable to argue, and we've argued it, that the tadpole stage may have been the version that gave rise to vertebrates or chordates. They lose it by metamorphosis for most of their life. So here's a colony that's going through separate stages, so you have to follow me. This is the one you see, okay? It has a bud that's growing next to it, and there's a little tiny bulge, which is the secondary bud, of stem cells and body wall cells. That grows so that after a week, it gives rise to this. That bud gives rise to that. And that grows for a week, becomes that, and then it dies, and it goes there, and then it dies, excuse me. So you have a 3-week lifespan of individuals, but we have colonies of those individuals in our laboratory at Hopkins Marine Station that have been there since 1981. So the genome is as immortal as we are, but the individuals have a 3-week lifespan. When 2 colonies or 2 individuals butt against each other, the blood vessels, the ampullae, push into the tunic of the other one. And then within 24 hours, they either form a line of immune rejection, or they fuse their blood vessels and become parabionts, sharing a blood circulation between 2 genetically distinct colonies of individuals. Here's fused blood vessels going across, and here's the rejection point just starting between those 2 colonies. These are the blood vessels that you can see that are in the tunics of either side. In 1982, we published, Jenny Schofield and I, that this rejection or fusion was due to a single gene locus that had over 100 alleles in Monterey Bay, as many alleles in the Botryllus, that's the name of our organism, as there are HLA alleles in humans in this room or H2 alleles in mice. They're all major histocompatibility complexes. We thought at that time that the gene that encoded their fusion or rejection by immune recognition would be the forerunner of ours, but it ain't. But the rules are kind of interesting. So if this is AB and AB at that single locus, they fuse. If they don't share an allele, they reject. If they share one but not another, they fuse. Now, if this had been T-cell mediated, the AB colony would have seen C elements that were foreign and rejected. So this is exactly the behavior of an innate immune system that we have in our bodies called natural killer cells. Natural killer cells see self and stop killing. This organism 550 million, derived 550 million years ago, doesn't. So now, if they fuse, if you think about it, with over 100 alleles, the only ones that between colonies will fuse and have orange blood cells cross into this formerly purple colony and now inhabit the body through the shared circulation have to be kin. So within your family, you'll find somebody 1 in 4 who's identical to you, but you'll find many, many who are haplo, that is, one allele of HLA plus one HLA not shared. But it means it is only shared among kin. And why is that important? We looked at the shared cells. Now, this is 2 colonies, orange and not orange. We're looking from the underside. And what we can see is this is the stage when those invertebrate stages are now forming testes and eggs for the next round of reproductive development of blastula, gastrula, neurula, and tadpole. And they share a blood circulation across. So we could put those together, let them fuse, and now look at allelic DNA markers to see when a new bud comes up, is it derived only from the one from which it bud? Or are there circulating cells, circulating stem cells, that in this parabiotic pair can land in the niche and compete for niche? These experiments were done by Doug Stoner, who sadly died a couple of years of glioblastoma. But here's one of the papers that we published now 19 years ago. So that was colony 1, colony 2. This is the blood, the allelic markers of A, B are here. This colony has only A markers, okay? These are DNA polymorphisms of dinucleotide repeats. We're looking at bud, sperm going from the outer edge to the inner edge. And there's one thing that everybody should see that's an anomaly. Anybody want to guess? No anomalies? All of these are A. Over here, you see some things that are A. And this is always under where S is, sperm. So although they share soma, and the blood is chimeric on both sides, all the germline comes from colony 1. We tested whether this was heritable. We did it many, many times. 2 different papers. So here we have colony F and colony B. Now, because there's so many individuals in it, you can cut the colony into pieces, and you can put them together many times to see genotype 1 and genotype 2. So in the germ phenotype, always F beat B in all the individuals in both sides of the colony. But it didn't beat it in the soma. Now, this mixture tells us we have to go look for different somatic stem cells, like blood stem cells, heart stem cells, brain stem cells, gut stem cells, and so on. When we do another test with G and B, G wins both the germline and the soma repeatedly. So we know that F beats B in the germline, G beats B. So when we put F together with G, it beats them, although G beats them in the soma. And when we make trichimeras, because you can put 3 of them together, the germline winner is always the same. So this is at least asexually inherited in the daughter cells of the individuals themselves. So there is a competition for germline niches. I'm not going to show you the data. We isolated, Diana Laird and I, stem cells from the colony. We transferred them from a winner to a loser, and even a small number of stem cells from the winner won the germline when the loser developed gonads. So we have long-term somatic and germline chimeras, and these are caused by parasitic lineages of stem cells. It is heritable who is the germline winner and loser. And I didn't tell you that's not only through asexual inheritance, but we went back to the breeding records in our mariculture at the Hopkins Marine Station. It was also inherited reproductively. We don't know the Mendelian genetics of it. We have not isolated the genes of it. We're just getting back to that now. That was 1999. Here's the important point. By having highly polymorphic histocompatibility, you prevent those predatory stem cells from invading your developing germline unless they come from kin, ones that share alleles with you. Now that leads to a very difficult proposition. When you're talking about doing Hardy-Weinbergs of selection, obviously when F had the germline and G the soma, the body and its selection for survival in the ocean was only related to the germline it would pass on to the next generation by the 50% of genes that are shared between kin. And every time I go ask Marcus Feldman, the guy who really is supposed to tell us all about this, to write a Hardy-Weinberg for this or anything, he says, sure. Then he doesn't answer my emails for months. It's too hard for them. But what this does say is that natural selection acts where the units of natural selection can be the colony, the individual, or the stem cell lines. And of course, Richard Dawkins would add gene, but we'll see. So just when I felt good about this massive discovery that we've discovered multiple levels of units of natural selection, one of my students pointed out the log of the Sea of Cortez that John Steinbeck went with Ed Ricketts gathering samples in the Bay of Cortez off the Gulf of California. And he wrote his observations in a little book called The Log from the Sea of Cortez. He said, there are colonies of pelagic, that's seagoing, tunicates, which have taken shape like the finger of a glove. Each member of the colony is an individual animal, but the colony is another individual animal, not at all like the sum of its individuals. So a man of individualistic reason must ask, which is the animal, the colony, or the individual? You could just say, instead of animal, the unit of natural selection. And he answered, he said, both. So he had the fundamental insight. And right after that, I called a novelist friend of mine and cursed him out. So we wanted to test whether there were germline competitions in we as individual organisms. So we tested it in mice. Hiro Ueno and I developed mice whose embryonic stem cells expressed either red, green, or blue fluorescent proteins. They inherited it. This is just growing them together. So we put them together into the blastocyst of an uncolored mouse. So now we have four colors, uncolored, red, green, blue. Now we let them develop. And when we look at the intestines, immediately we can see something that was never immediately apparent. You follow this crypt to tip. The crypt determines the clonal precursor. All blue. Every cell is blue. If there were mixtures of stem cells in the gut, intestinal stem cells, they would be mixtures for life. But they're not. Every single crypt. And then we cut here and we see that dozens of them, adjacent ones, came from a single cell. So we knew already that you could study clonal organization from stem cells in any tissue you wanted with this method. And we're going to try to make a super one that has more than four colors, because many tissues you'd like to trace have more than 4 kinds of cells that they make. But when we looked at the germline, so we dissected the testes of the males, we found first, this isn't on this slide, that the left and the right testis had exactly the same colors, but never, or almost never, all 4 possible colors. So the blood will be 4 colors, the skin will be 4 colors, but the testis is 2 or 3. And this tells us how many cells separated in early development to give rise to the germline stem cells. And the only number that fits by Poisson distribution is 4 cells. And Turnbull and Ueno figured that out, and then they told me. So you see large regions, just like in the intestine, of adjacent seminiferous tubules that are a single color. But here's the shock. When you look at them during the development of the testis, P0 up to P14, that's birth, 14 days after birth, every single seminiferous tubule is mixed with the colors that it has. So you go from 14 days after birth, 2 weeks before they become reproductively competent. This is before meiosis sets in the germline. You have mixtures of cells. But, as we noticed, between day 14 and 21, about 99% of the germline cells die. And the ones that are left give rise to the 2 or 3 colors that are in the testis by adjacent. And they're at the level of the germline that got in there because they are the left and right testis is the same colors but not the same distribution of clones. So that's powerful. It says existing within us is a selection system, or at least in mice, where most of the germ cells that are being made are being culled, and they're dying. Now, I'll just say, and this is speculation until Aaron Newman tests it, that this is the time in mouse development when the invading retrovirus elements called retrotransposons are moving from one spot to another within a cell. They leave a repaired spot and they have a new spot coming in. It happens all over the place. There are genes that are used to try to regulate the consequences of it, Pee-wee and me-wee and so on. But what we're saying now is that probably all of those ones that are culled had suffered genetic damage, so they could not be functional sperm. And fitting with that is that when we express the anti-programmed cell death gene in spermatogenesis, BCL2, they make sperm, but they're infertile. So that the vast majority of the ones that died probably weren't good. Now, what does this mean for evolution? Well, just like in the tunicate, there are new rules here. If retrotransposons are moving, they are changing the genes way faster than all of the geneticists told us cosmic rays would give rise to single nucleotide polymorphism. So we need to know if this is general, if this is true in humans, and we need to know if the sperm in BCL2 that are infertile have sustained damage and the others not. Then we would have to think about cell intrinsic pathways to know for every gene in the body, is it functional or not? Now, we immunologists know that in order not to get autoimmunity from T cells, a part of the thymus called the medulla has a gene called AIRE that opens up all genes of the body, whether thymic, brain, thyroid, or other, to express the protein, presumably so that all self-proteins are seen by developing T cells, and those T cells that happen to have autoimmune receptors against self are told to die and are eaten. So this could be incredibly important to understand evolution and how the invasion of retrovirus elements that can self-replicate once they're in a cell, once they've invaded the germline, are used for their own use, of course, to spread, and maybe, we'll figure out, are used by the hosts. So going back to Botryllus, we proposed back in '96 that you could have equal competitor stem cells, you can have super competitor stem cells, and the ultimate of that would be a malignancy. And if you didn't have a histocompatibility gene in the tunicate, the malignancy would spread through the ocean and the species would be extinct. It drives genetic polymorphism in the species, not only of the histocompatibility gene, but by keeping them away from invasion, it drives them for being individuals that can be units of natural selection. Now, going back to the blood-forming system, we want to look at whether this happens and whether the development of malignancies could be coming from competitive hematopoietic or blood-forming stem cells. And this is just to show you that we, over the years, have been able to identify, at least in mice, almost every step of hematopoietic development from the stem cell. In the first step, they lose self-renewal, but each cell, the multipotent stem cell, can, when transplanted, give rise to all types of blood, but only for 8 weeks, because it doesn't self-renew. As we go down, we have each of these cells, and each of these cells have their own gene expressions. All of you can go on that. You should write that down, HTTP, right? Gene expression. Because you can go to that site, everybody, and for free, in hematopoiesis and in some other systems as they come on, say, I'm interested in a gene called c-KIT. And it will tell you the exact quantitative level of c-KIT expression on every one of those cells. Now, the point I want to make here, now that you've all written this down, and you can talk to Jun Seta about it, is that there's no self-renewing cells in myelopoiesis other than the blood-forming stems. So we wanted to look at human acute myelogenous leukemia. And we've been doing this since 1998, when we first set up a mouse system to get acute myelogenous leukemia. And when David Traver in 1998 looked at the genes expressed in the leukemia cell that we could transfer from a donor to a host and transfer the leukemia in mice, we found those genes were involved in known oncogenic lesions like BCR-ABL, AML1-EDO, JAK2 kinase, and so on. But we also had anti-programmed cell death. We also had loss of tumor suppressors. We also had telomere elongating. That is a whole bunch of genes. And if you look carefully at each of those genes, none of them endow self-renewal. So how can a normal cell turn into a leukemic cell if all these genetic changes are not about self-renewal? The obvious hypothesis to us was that each of the changes would occur in a self-renewing cell, and that if they occurred downstream, they wouldn't cause leukemia. Okay? Everybody in this room has BCR-ABL transcripts. You had a translocation. You got BCR-ABL transcripts in your blood. Almost none of you, or maybe none of you, have chronic myelogenous leukemia, the leukemia that can derive from BCR-ABL. That's because they're in cells that have a finite lifespan. They cannot build up a clone that has enough mutations to now make it transition to self-renewing, spreading behavior in the body. So here are a list of genes. Now at the very end, we actually had shown in mice, and then Katrina Jameson and I showed in humans with chronic myelogenous leukemia that the last mutation appears to be one that does cause proliferation and therefore could cause self-renewal, the Wnt beta-catenin pathway. So we proposed that you would have clones of hematopoietic stem cells that eventually would give rise to the leukemia stem cell. In human AML, they're almost all at the multipotent progenitor. In the transition to myeloid blast crisis in human chronic myelogenous leukemia, it's the granulocyte monocyte progenitor. Those are the only cells that transfer the leukemia from humans to immune deficient mice. So we decided to investigate this. We being primarily Max Jan, who was a shared graduate student between me and Ravi Majeti. Ravi Majeti, an independent assistant professor. Ryan Corsa Zimmerman, who's still a graduate student in Ravi's lab and is still carrying on this work. Tom Snyder in Steve Quake's lab to help us do the genomics. So the experiment that we did was to take human AMLs and with exome sequencing, catalog the mutations. And now also do exome sequencing of T cells from the same patient so that we would be looking at variations that were somatic variations rather than allelic between individuals. We get rid of all those. Here's patient 70. Here's all the mutations. So Max Jan and Ravi and Tom and I watched, built DNA primers around each of the mutations that were unequivocal primers. And from the same patient, who happened to be in remission at the time but is dead now, took hematopoietic stem cells and singly deposited each hematopoietic stem cell, grew it up into a little clone, and tested with all the probes to try to get at N mutations in the leukemia, N-1, N-2, N-3, N-4. So you could not only get the mutations, but the order of the mutation. And here's 3 of the cases. Now together the labs have done about 21. The first group we did had an identifying indel called the FLT3 internal tandem repeat. So there were DNAs around that. And when I first asked hematologists, would FLT3 be driving it and the other mutations be added on and then you get a leukemia? Or would FLT3 be the last one? It was pretty even, but most of them said it'll drive it. It drives the proliferation of the clone. And then you pick it all up. In every leukemia, it's the last event. So the order will make a difference. In this patient, the first mutation, first clone, had one allele of TET2 with a stop codon. In that patient and in another patient, the second mutation was another allele of TET2. Now, how could we see these? At every step, every mutation gets a selective advantage for that cell over normal hematopoietic stem cells. That is, they're competing. They don't increase the total frequency of blood-forming stem cells. They compete. Myelodysplastic syndrome. You have mutations that now 95% of the cells are derived from a single clone that had monosomy 7 or 5q minus. It's not a leukemia. It's a disease. And those early stages aren't leukemia. They're leading to the disease. Okay? CTCF, FLT3 last. Now, FLT3 didn't occur at the level of purified hematopoietic stem cells. There were no stem cells that had added the last FLT3 in any of them. And when Ryan and Ravi went back and they looked at NRAS, KRAS, none of those were the driving ones. They were they were in the last step in the multipotent progenitor that drove the clone. So these genes, TET2 is the enzyme that helps demethylate cytosines in methylcytosine in DNA. And as you know, in order for a gene to go from being not expressed to expressed, the islands must go from CpG methyl to CpG non-methyl. There's enzymes that do it. These guys can use the product of 2-hydroxyglutarate for the oxidation reaction, and that too is a mutation called IDH1 or IDH2. Most of the early mutations change the ability of the cell to go through the normal epigenetic changes of differentiation, yet they still proliferate. Okay, so what I've told you, and I'm near the end, is that in AML, the progression, the pre-leukemic progression, is in a blood-forming stem cell. But the leukemia stem cell is a daughter of that clone at the multipotent progenitor or the granulocyte monocyte progenitor stage. There are no leukemias that we found that are leukemias of hematopoietic stem cells in mice or in humans. Remember, I told you got competition. None of the early steps, BCR-ABL, TET2, any of them, can increase the number of stem cells relative to the number of cells in the bone marrow. So whatever the real or virtual niche is for stem cells to self-renew, it is the most carefully regulated of all the steps. If this is true for leukemia, it can be true for all cancers, because cancers only derive from tissues, with the possible exception of the liver, and I still don't believe it, only derive from tissues that have normal stem cells that self-renew to give rise to differentiated progeny. Brain does. Most of you probably didn't know you had stem cells or cells still dividing at my age in your brain. So almost for sure, since most of the mutations that are cataloged in all the cancers are not endowing self-renewal, it'll be true in cancers. And we have to test all cancers. The last thing I'll say is that when we compared genes between leukemic and normal, we found CD47, the don't eat me signal for macrophages. That was expressed on the leukemia. Keep going, keep going. It's so slow. And just to make a long story short, my last sentence will be, we have made monoclonal antibodies that block the don't eat me signal. All human cancers have CD47. All can be transplanted with more or less efficiency into immune deficient mice. All of them are susceptible to anti-CD47 therapies. This is just a list from a year ago of all that we had tested in vivo, human tumors in mice, primary tumors. And the final slide is that our antibody, made by academics in an academic study, funded by the California Institute of Regenerative Medicine, are in clinical trials. The solid tumor trial is going on here at Stanford now to find safe and then effective doses. And the acute myelogenous leukemia trial will start in June in the UK. Thank you.
Copyright Stanford University, all rights reserved.